Hi, everyone, and welcome to the ARTNet podcast. I'm your host, Dr. David Goodrich from Roswell Park Comprehensive Cancer Center and the ARTNet Data Coordination and Management Center. Today, we're joined by Dr. Vlad Sandulache from Baylor College of Medicine, where he holds appointments in the Department of Otolaryngology, Head and Neck Surgery, and the Department of Radiation Oncology. He also leads a research program at the Michael E. DeBakey VA Medical Center in Houston, Texas. Last podcast, we talked about acquired treatment resistance, something that develops after therapy begins. The tumor is exposed to therapy, adapts under pressure, and eventually finds a way to survive. But that might be the common experience for all tumors. Well, and what if that timeline is misleading? What if many of the biological programs that determine treatment success or resistance are already active before the very first treatment is delivered? Rather than asking how tumors become resistant, today's conversation asks a different question. How do tumors arrive already prepared to resist therapy? That question sits at the center of much of Vlad's work. Today, we'll discuss how different exposures like tobacco, environmental stress, metabolism, immune interactions can shape tumor biology long before treatment starts and influence how those tumors respond to that therapy. We'll also talk about how understanding these early events may allow us to identify high-risk tumors before they fail therapy and ultimately design more effective treatment strategies for such tumors. Vlad, welcome. We're happy to have you join us today for the ARTNet podcast.
Thank you very much. Pleasure to be here.
Vlad, so you study a different type of cancer. What drew you to study head and neck cancer?
So I'm a head and neck surgical oncologist at Baylor College of Medicine. I think I became interested in head and neck cancer probably as a late medical student when I was completing my doctoral work and medical school work at the University of Pittsburgh. And I would say what drew me to the disease and more importantly, the patient is that head and neck cancer, frankly, is just really a miserable disease. Leaving aside the fact that it's deadly, no patient who gets head and neck cancer is ever the same after that initial diagnosis, even if the cancer is cured. So much of what makes us human beings, what we use to define ourselves as people, resides in the head and neck region. The way we look, the way we talk, the way we speak, the way we eat, the way we breathe, the way we interact with other people. When you have head and neck cancer, that fundamental reality changes, even if the cancer is cured. So it's really a very challenging disease for the patients, and I think that makes it a challenging disease for the oncologists that treat it. I think the other part of it from a scientific perspective is that 45% of our patients fail conventional treatment, and we really lack a very good understanding for why that happens, how that happens, and more importantly, we're pretty limited in what we can do about it. So I think it's a disease that's easy to become sort of impassioned about as a clinician, but it's also a biological question that's pretty much in your face if you're interested in cancer biology.
Right. So the cancer you study is, of course, very different from the myeloid malignancies that we discussed in the last podcast. What are some of the unique challenges in studying this cancer that might make it different than studying something like acute myeloid leukemia?
So if I think back to my days as a postdoc or during my residency, there was a lot of excitement. We were sequencing both solid and hematopoietic malignancies. Those were the early days of the TCGA project. We were doing whole transcriptome RNA sequencing. And there was a lot of hope that we would find both biomarkers of disease processes, but more importantly, actionable biomarkers, points of intervention, whether those be oncogenes or tumor suppressors that we could target. And I think, obviously, there was tremendous success for things like lung cancer with EGFR inhibitors for EGFR mutant lung cancer, ALK inhibitors for ALK mutant lung cancer, BRAF inhibitors for BRAF mutant melanoma, and thyroid cancer. In contrast to even those solid tumors, head and neck squamous cell carcinoma is essentially a desert of actionable genomic, transcriptomic, or proteomic events. So there's just not a whole lot of simple stuff to target. And the things that we could target, usually the targeting ends up being not very effective or very toxic. So today in 2026, the backbone treatment for head and neck squamous cell carcinoma is pretty much the same as it was 25, 30 years ago, a combination of surgery, radiation, and cytotoxic chemotherapy. The most commonly utilized cytotoxic chemotherapeutic agents are platinums. And of those, cisplatin is usually the backbone for the vast majority of our clinical trials and for the vast majority of clinical practice. So it's a tough disease. It doesn't give you a lot of opportunities to be clever and find that one TKI or that one small molecule inhibitor that's going to give you significant cytotoxic activity.
So, you're using therapies that have been around quite a long time. You don't have a lot of actionable targets that you can treat patients with if those therapies fails. And you mentioned that 40% of the patients that you do treat with those therapies do fail. So can you give us some perspective on the scope of that problem and why it remains such an important clinical challenge and an unmet need?
One of the things I tell all my patients is that the reason we spend so much time planning for their initial treatment and discussing their initial treatment regimen as a multidisciplinary treatment team that involves radiation oncologists, medical oncologists, surgical oncologists. The reason for all of that preparation is because our first chance to treat and cure head and neck squamous cell carcinoma is our best. Recurrent metastatic head and neck squamous cell carcinoma is almost universally fatal. And the reason for that is because we don't have really good second line options. Surgical salvage after radiation based failure is seldom effective and only in a minority of patients. Immune checkpoint inhibitors will demonstrate somewhere around 15 to 20% response rates, but the durable response rates are in the low single digits. So it's really that first chance at treatment that's your best chance to affect the cure. And after that, the salvage options are extremely limited.
Yeah, so you have one chance. Um, and during your years of treating patients, you noticed something that didn't quite fit the acquired resistance paradigm, where some patients or some tumors seemed almost destined to fail from the very beginning. They offered almost no response to the therapy, well, whereas other patients responded quite well. And since you only have that one chance to cure, obviously some patients are going to do much, much better than others. So what observations led you to believe that treatment failure often reflects biology that already existed before the therapy even started?
So I think anybody that's treated a head and neck cancer patient in the last 20 years has seen two very distinct phenotypes. Probably over the last 10 years, more frequently than in the past, we will see patients whose disease is associated with and we think driven by human papillomavirus, so what we call HPV positive head and neck cancer. And we've seen those tumors melt away. You'll see somebody who comes in with a bulky tumor at the primary site and regional basins, and they get to 20 or 30 gray of radiation, which is nowhere near enough to kill a squamous cell carcinoma, but might be enough to, let's say, kill a lymphoma, and the tumor's gone. Obviously, that's an extremely radiosensitive tumor. On the other hand, we've seen patients that come in with bulky disease. And because the disease is bulky, because we worry about the risk of distant metastasis, we'll often start with something called neoadjuvant or induction chemotherapy. And the most aggressive regimen of that is the 3-drug cocktail that includes platinum, taxane, and say 5-FU. So it's an extremely rigorous and toxic regimen. And you'll see patients whose tumors progress during neoadjuvant or induction TPF. Obviously, there's biology there. That there's a clear and distinct sensitivity to cytotoxic chemotherapy and to radiation, both of which obviously converge on significant levels of DNA damage. So there are clearly tumors that start the treatment with a predisposition to really not care that much about it. They are clearly loaded for bear, ready to resist DNA damaging agents. And there are other tumors that are going to be very sensitive.
Right. So researchers often talk about that innate resistant, the resistance from the very beginning, versus acquired resistance, those that might evolve resistance over time. And we think about them as completely different processes, but you've suggested this distinction may actually be misleading. Why do you think the boundary between innate and acquired resistance isn't as clear as we often assume?
Yes, that's a great question. And honestly, that this is something that I probably thought about more and more over the last 4 years or so. And a lot of it is due to the conversations that we have within the context of ARTNet, which span multiple diseases, multiple specialties, highly variable biology. We start treatment because the payer decided that this is the time to start the treatment because the patient decided that this is the time to walk into the hospital or the clinic, and because this is the time when all of the testing that was required to start treatment was complete. It's purely an administrative time point that has nothing to do with tumor biology or evolution or some sort of insight into what's happening in the tumor. We just decided to treat the patient on Monday because that's when we were ready to start the radiation. Um, First of all, this distinction between innate and acquired is really based on an operational time point, not on anything biologic. So that made me think that there may be things that are happening before the official start of treatment that the tumor is experiencing, which are going to put it in a position to be more or less sensitive to that treatment. And um, there are some things that are unique to head and neck cancer, or at least a little bit more specific to head and neck cancer, but there's something that's not unique at all to head and neck cancer that's broadly applicable across both solid and hematopoietic malignancies, and that is a tumor's ability to evade the immune system. So if you think about it, every single patient that walks in through a clinician's doors has a tumor. That tumor started out as a pre-malignancy. It evolved into a malignancy. And for it to be there, for us to see and biopsy and treat, clearly it has been able to partially defeat the immune system over a prolonged period of time. So there are cytotoxic T cells that are constantly trying to come into that tumor, kill the tumor. The tumor's fighting them off. Now, the tumor is not trying to be resistant to immune checkpoint inhibitors, but it will, by definition, be partially resistant to immune checkpoint inhibitors because it's been fighting the cytotoxic T cells throughout its entire life. So it's natural to understand that there are forces that are being exerted on tumor cells during their development that are going to be there during treatment. So the overlap between those forces and the treatment is going to be different for different diseases. So for example, whereas immune evasion is obviously universal across cancer, in our case and in other solid tumors, it may be something like exposure to oxidative stress. And obviously, if you've been conditioned by long periods of pre-existing oxidative stress and the treatment that you're going to use for that cancer is dependent on oxidative stress, you could easily see how this division line between innate and acquired resistance really starts to become blurred.
So I guess what you're saying is that tumors evolve from the very beginning, and when treatment begins, is a little bit arbitrary depending on when the tumor is diagnosed. And so there can be a lot of evolution in the tumor that occurs before it's even recognized and before it's even treated. And perhaps some of that evolution is preparing the tumor to deal with stresses it might encounter with therapy. So if evolution can begin before treatment, something must be shaping those tumors ahead of time to prepare them for resistance. So, what kinds of selective pressures have tumor cells already been living through that you think prepare them for resistance to, let's say, cisplatin and radiation?
Immune evasion is one of those things that I think we've been thinking more and more about over the last 10 years, certainly the last 5 years, given the advances in immunotherapy. But if we go back to work from 1990s, early 2000s, um, it became pretty clear once we were able to deploy both large scale, broad based metabolomics approaches and more precise carbon based methods of interrogating the metabolic state of the tumor. That tumor metabolism is fundamentally unbalanced. The tumor has to do a lot of things which are not normal. It has to proliferate at extraordinary rates. It has to generate biomass in order to support that proliferation. And that's not really what eukaryotic metabolism was designed to do. It's kind of like taking a Honda Civic and basically putting your foot on the gas and revving the engine at 7,000 RPM. I mean, that works up to a point, but if it stays a Honda Civic, then that engine's going to break down. Tumors adapt to that, but they adapt to that in ways that are not always completely inert. So, for example, if you try to pump a lot of carbon through the mitochondria, you're going to generate excess reactive oxygen species because the mitochondrial electron transport chain is leaky by design. And that's one of those sources of oxidative stress that's often overlooked. Every single cancer cell is sitting on a knife's edge where they're trying to generate enough energy and enough biomass by running the mitochondria and glycolysis and secondary pathways at maximal speed, but at the same time not generating enough intrinsic reactive oxygen species to where they self-destruct. So because the tumor is in this constant state of metabolic adaptation to its proliferative needs, it's slowly poisoning itself with reactive oxygen species. Now, in the process of doing that, it has to adapt at the same time. So you can easily see how you would have a vicious cycle from the perspective of the patient, but a very favorable cycle from the perspective of the tumor, to where the more the tumor can perturb its metabolism and adapt its metabolism to support high rates of proliferation, at the same time, the more it becomes adaptive to endogenous oxidative stress.
Why would oxidative stress in particular prepare a tumor cell to survive cisplatin or radiation therapy? Is it based on how those therapies work?
So, yes, the simple answer is radiation kills tumor cells, or any cells for that matter. Basic radiobiology teaching is about 2/3 of the toxicity of radiation is driven by oxidative stress. Free radicals, 1/3 is driven by direct DNA damage. Cytotoxic chemotherapy sort of depends. Certainly, it does cause oxidative stress, but the easier way to think about it is cisplatin. Cisplatin in and of itself is a free radical. So when your primary non-surgical treatment modalities are either 2/3 driven by ROS or are in fact themselves a free radical, certainly pre-existing adaptation to oxidative stress is going to have a deleterious effect. Um, the other thing to keep in mind is that outside of the fact that the tumor cells themselves are bathing in high levels of ROS just as a result of their metabolic flux, the patients will often be bathed in oxidative stress depending on environmental exposures. There's a strong body of literature in the context of lung physiology and lung cancer demonstrating that smoke generates oxidative stress, cigarette smoke. We've demonstrated the same thing in our patients and our preclinical murine models of head and neck squamous cell carcinoma. So certainly smokers who have been exposing their premalignant lesions, which then turn to a cancer for months and years before they ever present for treatment, are going to condition those tumors to much higher levels of ROS. Even if you move outside of individuals that smoke cigarettes, air pollution will generate oxidative stress. I work at the VA. One thing that we continue to see patients with exposure to is Agent Orange. Agent Orange obviously was used extensively during Vietnam. People think of it as unique to that conflict, but it's not. The active compound in Agent Orange is dioxin. Dioxin is essentially a weed killer, and it's present in a wide variety of compounds in that family. It's an extremely effective free radical. If you look at the exposome in aggregate, there are going to be solid tumors where the exposome is going to play a pretty significant role in preconditioning those tumors to resist oxidative stress.
So when you talk about preconditioning, it sounds like that stress is almost like a training ground for cancer cells. And I'm wondering, how does that work? Do you think it's an evolutionary process? Do you think all the cancer cells are trained, or do you think some are stronger than others and you get sort of natural selection of those cells and then they're better prepared for the therapy? Or how do you think that works?
Great question. In a way, that's the $1,000,000 question. Whether or not the evolution that we see in solid tumors is a function of individual clones adapting or simple clonal extinction and expansion. And we can't really answer that question in patients, certainly not in head and neck cancer. What we've seen in preclinical models that if you take even cell lines, so cell lines are fairly specific, we assume some degree of homogeneity. When you expose these cell lines to prolonged periods of oxidative stress, whether that's exposing them to low doses of radiation, chemotherapy, or our preferred method, exposing them to cigarette smoke, which is what we see in most of our patients, we see both phenomena. So we have seen from what we assumed were isogenic, completely homogeneous cell lines, distinct clones appear with mutations in pathways that we could never identify even with pretty high resolution sequencing in the parental population. Now, you could say, Vlad, those mutations developed during your treatment, but it's somewhat unlikely over a reasonably short period of time. It's much more likely that what we're doing is we're uncovering very low frequency clones. And you might say, why were those clones such low frequency? Well, clonal extinction and expansion is going to be based on the selective pressure. If the only selective pressure is, can you grow fast in a plastic dish, there may be no benefit to activating certain pathways. If now you have to survive under conditions of oxidative stress, then obviously that changes the calculus from the perspective of the tumor and what's advantageous and what's disadvantageous. But we have also seen wholesale transcriptomic reprogramming of tumors to resist oxidative stress, even in the absence of mutations. So what we see is that when we expose head and neck squamous cell carcinoma models to oxidative stress, we see a conversion towards transcriptional pathways that are designed to rewire metabolism to resist oxidative stress. If there are mutations to uncover, they'll uncover those mutations and leverage those mutations. If there aren't, then they seem to execute a transcriptional shift in that direction, and they all get to the same place.
So let's talk about that transcriptional shift a little bit. If oxidative stress that tumors are exposed to during their evolution before they may be treated or even diagnosed is the training ground that helps them prepare to resist treatment, what do you think is the key pathway or key transcriptional pathway that allows them to do that?
Well, like any cancer researcher, at some point, I have to discover a pathway and and assume that this is the pathway. Being as fair as one could be, from what I've seen over the last decade, in terms of our work, the work of our colleagues, in terms of head and neck squamous cell carcinoma, I think our best candidate for a critical driver of adaptation to oxidative stress is NRF2. One of the reasons I believe that is because it seems to come up over and over again using orthogonal approaches. You see NRF2 hyperactivation when you look at tumors that have survived radiation. You see NRF2 hyperactivation being common in smokers. Smokers, not just with head and neck squamous cell carcinoma, but smokers with lung cancer, bladder cancer, and other smoking-associated malignancies. So I think when you see a pathway come up in multiple settings, especially when you're not looking for it, then you tend to think that it's more of a signal rather than an artifact.
Does NRF2, does that function in normal healthy people as well, or is that unique to cancer?
So that's what I think really helped convince me that we weren't on the wrong track. This is a basic redox sensor. The trigger for NRF2, which is a transcription factor to translocate, go to the nucleus, and turn on a whole bunch of genes, is literally oxidative stress. And that works through this beautiful chemical mechanism where a free radical will oxidize another protein called KEAP1. KEAP1 will no longer be able to bind NRF2, and then NRF2 escapes, goes to the nucleus, and turns on all these genes. This is as simple as a molecule of ROS appears inside of a cell immediately adjacent to the keep one protein, oxidizes the keep one protein, and then NRF2 is off to the races, which means the whole process is almost instantaneous. NRF2 will turn on the survival machinery required to survive oxidative stress as instantaneously as is biologically possible. And because of that, it works extremely well for normal tissue. So, for example, if you just poisoned yourself with a large dose of free radicals, this is exactly the mechanism you want maximally active in your kidneys and in your liver and in your lymphocytes, because the faster the mechanism gets turned on, the less likely you are to kill yourself and destroy your organs. In cancer cells, what they're trying to do is find the simplest, lowest energy solution to a problem. So NRF2 is the simplest, lowest energy solution to the fundamental problem that a cancer cell has, which is, how do I survive reactive oxygen species? So when it's simple and when it's low energy, a lot of cancer cells are going to evolve towards the same pathway.
So let me see if I understand this correctly. Exposures to stress during normal life might prepare a cell or an incipient cancer cell or an evolving tumor cell to upregulate this NRF2 keep one pathway in order for those cells to survive that stress. And that trains them to survive the stress of therapy. So let's connect back to the therapy that you use for head and neck cancer. How does NRF2 activation affect response to cisplatin, for example, or radiation?
So what we see in cancer cells where this pathway is hyperactivated is really a wholesale reprogramming of tumor metabolism. If you think about a cancer cell, it really only has to do one thing, which is proliferate. To proliferate, you have to generate biomass and energy designed to make two cells out of one cell. When you're exposed to oxidative stress, you have really two ways of dealing with that. So the conventional thinking was that a cell is going to let the ROS damage the DNA, and then it was going to become really, really good at fixing the damage. If you think about that, like from just a simple-minded perspective, that is really the highest energy and most complicated approach to dealing with DNA damage you could conceive of. It's kind of like thinking that you're basically going to let the water come into your house through a bunch of holes, and then you'll just figure out a way to get the water out of the house in the middle of the flood. And the solution to that problem is you patch the holes in the wall so the water doesn't come in. So what we see with NRF2 is the simple answer to a cell dealing with oxidative stress, which is that it builds up massive antioxidant defenses. So what does that mean? Well, glutathione is a tripeptide. It's one of the primary redox of forces inside of the cell, because the molecule of glutathione will capture a free radical and instantly become oxidized, and that free radical becomes neutralized. So if you double your glutathione reserves, then you're going to be able to survive a burst of ROS. And the nice thing about that is it's going to be instantaneous. And the faster the process, the better off it is for the cell, because radiation delivers intracellular ROS instantaneously on a physical chemical scale, right, within femtoseconds. So if your cell is literally preloaded with reducing equivalents like glutathione, thyrodoxine, and other thiol-containing moieties, then you're going to be able to neutralize that burst of ROS instantaneously. Whether it's a burst of ROS coming from radiation, whether it's cisplatin slowly entering the cell, as soon as it appears in the cytoplasm and the nucleus, it's neutralized. And NRF2 will switch tumor metabolism from this rapid proliferative state to this other state that's designed for survival. And it does so by making a trade-off between energy proliferation and biomass. And the reason for that is if you've just captured a whole bunch of molecules of ROS with your proteins, those proteins become damaged. They have to be recycled. They have to be regenerated, which means carbon flux can now no longer be used simply to go from one cell to two cells. Carbon flux now has to go into biomass regeneration so that cell can survive the next round of ROS. This is what we see in our preclinical models. And unfortunately for our patients, it's exquisitely simple and low energy.
And so, yeah, I guess one of the problems is any therapy that relies on oxidative stress to kill the tumor cell is going to be affected by this pre-training activation of this pathway. So if that's true, is there something we can do to intervene once you've identified a resistant tumor? What can we do about it?
Well, let's start with the negatives first. It's a transcription factor. So transcription factors are tough. They turn on a lot of genes. If you try to kill a transcription factor, you're going to have a lot of effects. And those effects are not going to just be on cancer cells. They're going to be on normal cells as well, particularly if this is fairly ubiquitous as it is. And if it's a critical response mechanism to oxidative stress and normal pressures inside of normal tissue. What does give us a window of opportunity is the kinetic profile of NRF2. So the fact that it comes on quickly and it turns off quickly is actually favorable. And what I mean by turns off quickly is NRF2 is constantly degraded. So it's one of those proteins in the cancer cell where the levels of the protein and the levels of the activity are not so much driven by the levels of transcription and translation of the protein itself, but how quickly it becomes degraded by ligases and proteasome and multiple other mechanisms. So that means that if there isn't something turning on NRF2, it will very quickly go away. And if you can switch the on-off mechanism to push it in the direction of degradation, the signal from NRF2 will disappear very quickly. Now, you might say, well, so then let's work on keep one. Obviously, this is keep one based degradation, but that's a little bit hard because we're talking about protein-protein interactions. And one of the mechanisms of NRF2 activation is you get activating mutations in NRF2, which prevent keep one from binding, or you get inactivating mutations in keep one, which prevent it from binding NRF2. But there are other mechanisms of stabilizing the NRF2 protein. And one of those mechanisms is phosphorylation. So when you start to look about at what things you might be able to inhibit in terms of NRF2 stabilization, phosphorylation of the protein does stand out. And that's helpful. And the reason that's helpful is because what we've been doing for 20 years, I mean, we as a field, is figure out how to inhibit phosphorylation events, right? Kinase inhibition has been a boon for a wide variety of other solid tumors, not so much for head and neck cancer. But in this particular case, all of the work that's gone into developing kinase inhibitors does actually help us. And the reason for that is because if you can block the PI3 kinase pathway, you can actually induce a fair amount of destabilization of the NRF2 protein, which isn't something that we expected right from the beginning, but it is something that we're seeing. That gives us a lot of hope that we might be able to target this pathway.
So you're using PI3 kinase inhibition as a potential way to modulate that NRF2 pathway in order to sort of downregulate it so the cancer cells are less prepared for the therapy you're going to give them. If that's the approach, obviously you have to be able to identify those tumors that are unlikely to respond to the therapy first, right? Can we identify those tumors before the treatment begins so that we could maybe undo their precondition training with oxidative stress to make them more sensitive to the therapy?
I think the short answer is yes. The one caveat I would have is that for quite a while, the field relied on simply looking at NRF2 and KEAP1 mutations to define that subset of solid tumors, whether it's head and neck squamous cell carcinoma, lung cancer, or other tumors, that might be well suited for an NRF2 targeting approach. Our data would suggest that if you only target the mutants, you're probably going to miss out on a fairly significant plurality of tumors that demonstrate hyperactivation of the pathway in the absence of mutations. So we think that the best biomarker for what we would call an NRF2 hyperactivated tumor is a transcriptional biomarker. So not so much looking at whether or not you have mutations in NRF2 and KEAP1, although obviously those tumors are going to be good candidates, but looking at the downstream genes, which are the essential effectors of the antioxidant response. And when you see that signature, you can be fairly certain that that tumor is going to be chemoradiation resistant. And The reason we're pretty comfortable in that assumption is because a lot of those genes are, in fact, enzymes. So if I told you that you have 4 times as much of an enzyme whose primary goal is to recycle glutathione, well, you don't have to extrapolate too much to think that that tumor is going to do pretty well when it gets a load of ROS in the form of cisplatin or radiation. So that's probably a tumor where you want to intervene.
So your most reliable marker is then NRF2 is a transcription factor. So you're looking for activation of the genes that it normally regulates. So what type of technologies do you think are going to help us do that routinely in the clinic going forward?
Good question. If we want to deploy basic transcriptomic approaches, whether it's RNA-seq, spatial transcriptomics, anything else you might want to deploy, you're going to need a piece of the tumor. For most solid tumors, that's a pretreatment biopsy. Most head and neck squamous cell carcinomas are amenable to biopsying the primary tumor itself because they're somewhere in the upper digestive tract. So you don't have to rely as much on a needle biopsy or a core biopsy as you might for a lung cancer or a renal cell carcinoma. Whether or not we can pick up some of the signature and circulating DNA, it's something that's under active investigation. But the other thing that we've done over the last almost decade or so is to develop non-invasive measures of carbon flux and oxidative stress inside of solid tumors by leveraging hyperpolarized MRI. That's pretty hard technology to work with and deploy at scale in human patients, although I think we're getting a lot closer to that step. The beauty of it is you can interrogate a solid tumor in space and time in a way that's almost as good as what you can do with a liquid tumor. So the technology is a little bit far away from deployment at scale, but a combination of circulating DNA, primary tumor biopsy, and metabolic imaging, I think can give us a pretty good and robust window into whether or not that tumor is likely to be resistant to chemoradiation or not.
Is there a possibility there's evidence of that oxidative stress preconditioning in a tumor that might exist in the blood that could be more easily sampled, or maybe, depending on the tumor, of course, could be urine or some other bodily fluid that's more easily accessible than taking tumor biopsies? Is there hope for that?
I think we do have some advantages in the context of head and neck cancer over other solid tumors in the sense of saliva can be sampled, and saliva has been successfully sampled for a variety of purposes. So, for example, you can detect HPV DNA in saliva and potentially try to figure out if you're looking at a pre-malignant or very small malignant tumor that's driven by a human papillomavirus. Certainly, you can interrogate the microbiome by looking at saliva, and depending on how the tumor is shedding DNA and how quickly that DNA gets chopped up by the enzymes in our saliva, you could pick up some of those markers.
Yeah, I'm going to switch gears a little bit. So you're part of the ARTNet network, and I'm wondering how being a member of that network and interacting with people who study the different disease sites, different types of therapies, different resistant mechanisms, how has that influenced the questions that you're asking in the lab and maybe the approaches you're using to answer those questions?
I trained both during graduate school, but especially during my postdoctoral years in labs and with people who really practiced team science. That's how I evolved as a scientist to think about team science as the first approach. So for me, being part of ARTNet was just a natural next step. And you have access to so many smart people who study multiple diseases from multiple vantage points with overlapping and sometimes non-overlapping skill sets. It's just a fantastic environment.
I mean, you've already described in your own work, you've mentioned oxidative stress, metabolism, immunology, radiation biology, genomics, and clinical oncology. Those are all pretty big fields in and of themselves. So how has the network helped you incorporate those perspectives into your own work? Is there any specific example you could give, or even applying what you've learned in head and neck cancer to other cancers?
I mean, one thing that I would say is you get ideas and you get hints, right? I have some fantastic collaborators, and I formed those collaborations through ARTNet. And if I think I have a hint of a signal in a T cell, then I don't really extrapolate too much. I go and I ask a T cell expert and I say, am I crazy about this, or do we think this is a real signal? And if I have a question about the dendritic cells, then I'll ask one of the experts in antigen presenting cells that I've become a close collaborator with through ARTNet. You had Jeff Tyner on for the last podcast, and he's an expert in both understanding the actual evolution of tumors over time and tumors in response to treatment, and how you can target those tumors in a rapid fire way using high throughput screens and using both molecular screens and drug screens. There's really nothing in common between the disease that I study and the disease that he studies, but there's certainly a lot that I've learned from his approach to how we can interrogate our preclinical models and some of our tissue samples to gain some of that insight.
Great. So I ask this question at the end of each podcast, and I'll ask the same question to you. Given what we've talked about today, where do you see head and neck cancer and head and neck cancer treatment in 5-10 years? You've said that we basically use the same therapies to treat this disease that we were using 15 years ago. Do you think that's going to be with us for a while, or do you think we're on the verge of change?
I look at head and neck cancer treatment the way I look at the history of cars. In 2026, a car is still a car. It still has 4 wheels and an engine / battery and a steering wheel, and it moves in a plane, just like it did in 1940 and in 1910. But I would much rather be a passenger in a car built in 2026 than in a car built in 1926. So what I think and what I hope we're going to see with head and neck cancer is not necessarily a fundamental wholesale shift in the way we treat the disease. I don't think the biology is going to allow for that. We're going to find ways to interrupt this adaptation or this pre-adaptation to where we can interrupt the adaptation of the cell, but not generate too much off-target toxicity that we've seen over the last 5 years, I think speak well to that possibility. And if we can do that, and if we can de-escalate the treatment for the patients with sensitive disease and escalate the treatment for the patients with resistant disease, I think we're going to see enough gains on the margins to where in 2 decades from now, having head and neck cancer and going through head and neck cancer treatment is going to be fundamentally a different experience for the patient.
Vlad, thank you very much for joining us and sharing both your science and the vision behind your work and, of course, your clinical practice. And thank you to our listeners for joining us for this episode of the ARTNet podcast.
Thank you very much.